Hello. This presentation discusses a field in biology called cell signaling, otherwise known as how cells communicate with one another. It may surprise you to hear that cells communicate with each other, but they do. They continually send out signals to other regions of the body to maintain homeostasis and help the body function. Some ways that a cell might respond to these signals that they receive from other cells would be to transport a substance into or out of its cytoplasm. Maybe the responding cell will increase ATP production. The target cell could contract, as in the case of muscle cells, or divide, or could differentiate, change, and mature. Here's kind of a cartoon that shows you ways that cells could respond, meant to give you a little levity here. A signal could cause a cell to divide, and so you've got this nice cell doing division. And in the other picture, there's a mature cell saying, you know, when I was a little stem cell, I didn't know what I wanted to be either. And the baby cell is saying, but I'm so confused. Doesn't know what he wants to differentiate into. So cells might divide or differentiate, mature in response to a signal. They could also start making a protein or perform some chemical reaction. Now we will look at how cells actually signal each other. There are two general methods by which cells communicate. The first method is called direct contact. The signaling cell and the recipient of the signal, or the target cell, are close to each other. In this case, the signaling cell usually has a signal embedded in its cell membrane. In this case, the signal is shown in pink in the diagram, and it's referred to as a ligand or a signaling molecule. The target cell has a molecule that binds to this signal. That molecule is a receptor, and is shown in green. The second method also involves a ligand and a receptor interacting, but in this case, the signaling cell secretes the ligand or releases it. The ligand then binds to the receptor on a target cell, just like before. Because that ligand is secreted, scientists call this second method chemical signaling, because the chemical is released or secreted into the extracellular fluid. And the ligand will diffuse away from the signaling cell to eventually reach a target cell that has an appropriate receptor for that ligand. There are variations in both methods, direct or chemical cell signaling. But first, let's look at the big picture. One purpose of cell signaling is to alert multiple cells and organs to change in some way to meet the body's needs. All the cells in your body must coordinate their activities in order to benefit the overall health of the body. For example, when you exercise, both skeletal muscle cells and cardiac muscle cells must behave in unison to keep oxygenated blood pumping to those skeletal muscles that are contracting so quickly. Therefore, multiple cell types need to receive the same signal. I'll call it the exercise signal, so that both your heart and your skeletal muscles are working together. It is also true that your body cells receive multiple signals throughout the day, but it may not be necessary or even appropriate for a specific cell to respond to every single signal. For example, it would not be helpful for the smooth muscle cells in your large intestine to contract at the same rate as your heart muscle cells do during exercise. Instead, the large intestine muscle should ignore the exercise signal. So this cartoon, shown here, shows a signaling cell in the center. The cell on your left has the appropriate receptor and can respond, but the cell on the right does not have the appropriate receptor, and so no response will occur. So this leads to our first central theme in cell signaling. The cell can only respond if it has a receptor specific for the ligand or the signal. We refer to this feature as specificity. That is, ligand and receptor binding are specific. A cell can only respond to a ligand if that receptor can bind to it, and not every receptor will bind to every signal. This picture depicts this characteristic. Notice that the cell has 3 receptors. They're shown as pink blobs. There are many ligands or signals outside the cell. A square, a triangle, a circle, and another shape that reminds me of a stamp that you would use on a stamp pad. If you look closely at the receptors, you'll notice that they all bind to the stamp shape. This cell cannot respond to the square, it can't respond to the triangle or the circular signals. So it's common for a cell to have multiple receptors that will bind to the same ligand. And it's also common, although not shown here, for cells to have multiple types of receptors so that they can respond to different signals. We will not be discussing different types of direct contact cell signaling. Instead, for the remainder of this presentation, we will focus on chemical cell signaling. And there are 4 types of chemical cell signaling that differ in 2 main ways. One way is where is the target cell in relation to the signaling cell. The second way is how the ligand gets to the target cell. So these 4 types are listed on this slide, and we will go through those 2 ways that they vary. So the 4 types are paracrine, autocrine, synaptic signaling, and endocrine signaling. They all are chemical cell signaling, but I think of them as pathways because they describe where the 2 cells are and how the signal gets through the body to the target cell. 2 of the 4 types are shown on this slide. In the first type, paracrine, the 2 cells are close to one another. In the upper right there. The signaling cell, labeled the secretory cell, is releasing a green ligand. That ligand does not have to travel a great distance to get to the target cell, which has receptors for it. In this case, the ligand will just diffuse from an area of high concentration to an area of low concentration. You can see that the concentration or strength of the signal would not diminish much because the 2 cells are so close together. In the second type of chemical cell signaling, autocrine signaling, the 2 cells are actually one and the same. I mean that the signaling cell responds to its own signal. This occurs in the immune system, which you'll learn about in A&P2. Again, the ligand diffuses away from the cell that secretes it, but again, it doesn't have to travel very far at all to reach the target cell because it's the same cell. The third and fourth types of chemical cell signaling are depicted on this slide. Synaptic signaling is called synaptic signaling because the junction between a neuron, shown in the diagram right here, and the target cell, any target cell, is called a synapse. So this region is called a synapse. Anytime a neuron is signaling to a cell or activating a cell, it's called a synapse. So this is synaptic signaling. The end of the neuron is where the ligand will be released or secreted. And the ligand that comes from a neuron is called a neurotransmitter. In my mind, since these two cells, the neuron and the target cell, are so close to one another, it's very similar to paracrine signaling. But it's referred to as synaptic signaling anytime a neuron is involved. Again, the neurotransmitter diffuses away from the neuron across that synapse to reach the target cell. The last type of chemical cell signaling pathway is called endocrine signaling. In endocrine signaling, the signaling cell and the target cell are typically very far apart from one another. The signal must travel through the bloodstream to reach its target cell, and there may be many target cells. Diffusion will still occur as the ligand gets into the blood or leaves the blood, but the majority of the distance traveled by the ligand is in a liquid in the blood. Any signaling molecule that travels through the bloodstream has been released by a gland is called a hormone. So in this fourth type of cell signaling, the ligand is called a hormone because it travels through the blood. Now we are going to look more closely at the molecular level of signals and receptors. You will learn that there are two main types of ligands and two main types of receptors. They're shown in a diagram on this slide. Now these two types of ligands and two types of receptors may be used in any of our four general pathways of chemical cell signaling: paracrine, autocrine, synaptic, or endocrine. So the pathway is not what's focused on here. It's just the interaction between the signal and the target cell's receptor. You will find this helpful, even if it seems tedious at first, because we studied the nervous system and the endocrine system in detail in A&P1, and this will become very relevant and it will make things so much easier for you. Okay, so the two types of ligands are based on whether they like water or not, meaning whether the compound is polar or not. So one type of ligand is hydrophilic, polar, and the other type of ligand is hydrophobic, or nonpolar. From your study of organic molecules, you may remember that any compound that is a lipid or a steroid is hydrophobic. But all the other types of compounds, proteins, carbohydrates, and nucleic acids, are all hydrophilic. Generally, ligands are, though, only going to be lipids or proteins. Now let's look at the two types of receptors. The two types of receptors aren't based on their chemical nature. They're usually proteins. Instead, the types are based on where they are located in the cell, the target cell. One type is a protein embedded in the cell's membrane. Therefore, it is called a membrane receptor. It also might be called a cell membrane receptor or a cell surface receptor, but it's the same thing. It's on the membrane. The other type of receptor is a protein located inside the cell, either in the cytoplasm or in the nucleus, and that's called an intracellular receptor. So the two types of ligands are hydrophilic or hydrophobic. The two types of receptors are membrane receptor or intracellular receptor. Now, for a ligand to reach an intracellular receptor, it must cross the cell membrane. Therefore, it must be a hydrophobic compound, a lipid, or a steroid. Ligands that do not cross the membrane, such as proteins, will have to bind to cell surface receptors. You may be feeling that we have already covered a lot of information, so it may be helpful to review what the important points are at this point. The first point is you should know what the two general methods of communication are that cells use. The answer is 1, direct contact, 2, chemical cell signaling. You should know the 4 types of chemical cell signaling, meaning the pathways and how close the cells are to one another in each of the 4 types. So paracrine, autocrine, synaptic, and endocrine signaling. And then number 3, you should be able to list the 2 types of ligands, either hydrophilic or hydrophobic, and how that dictates whether or not it can get across the membrane. Number 4, you should be able to list the 2 types of target cell receptors and where they're located. So a cell membrane receptor obviously is located in the cell membrane. An intracellular receptor could be in the cytoplasm or the nucleus. And then number 5 brings all of that together and asks you to understand which type of ligand binds to which type of receptor. A hydrophilic ligand binds to a cell surface receptor, whereas a hydrophobic ligand binds to an intracellular receptor. Now we are going to look at what happens when these ligands bind to the appropriate receptors. Let's first start with intracellular receptors. We're going to look at the mechanisms and actions when a ligand binds to an intracellular receptor. So this mechanism will involve a ligand that is hydrophobic. It has to be to get inside the cell. Some examples that might make this more relevant for you include cortisol, testosterone works this way in your body, estrogen, vitamin D, and thyroid hormone. So the receptors, since they're intracellular, are in the cytoplasm or the nucleus. Now, the focus is what happens when they bind. The most common effect is to regulate gene expression in the target cell. So the target cell is going to change what genes are transcribed. A gene will either be turned on or turned off. And what that means is it will either be transcribed so that a protein will be made, or when turned off, it won't be transcribed, so that protein that it codes for will not be made. Cortisol works this way. It's a hydrophobic ligand. It's also considered a hormone because it travels to the target cells by the bloodstream. It's made in the adrenal gland and then travels through the blood to many target cells. The target cell that I chose here is skeletal muscle cells and fat cells. Its effect most often is to cause the synthesis of enzymes that break down lipids or proteins. So if you think of cortisol or cortisone as a stress hormone, what's going to happen is your body's going to start breaking down lipids and proteins. To do that, the enzymes have to be made to break down lipids, to break down proteins. So cortisol And the picture is a pink triangle or purple triangle, and it passes through the membrane into the nucleus, all the way across the cytoplasm, in through a pore or across the nuclear membrane, and it binds to the receptor molecule, which is kind of orange, the receptor protein, it says. Together, they bind to the DNA. Now, this, with other factors that bind, will either turn on the gene or off the gene. In this case, it turns on the gene, and therefore the gene is transcribed into messenger RNA. Messenger RNA then goes out to the cytoplasm. The ribosome, shown right here, will synthesize the protein, and that protein will be an enzyme that breaks down lipids or another protein, a storage protein, a nutrient. Okay, so that was intracellular receptors. Now we're going to look at mechanisms or effects that happen when a ligand binds to a cell membrane receptor. Remember, this ligand will be hydrophilic. It can't cross the cell membrane, so it has to bind to a cell surface receptor. Some examples in your body include insulin, epinephrine, and acetylcholine. Lots of neurotransmitters also are hydrophilic. The actions of ligand cell membrane receptor binding are not as straightforward as in the previous case of intracellular receptors, because actions will vary depending on what target cell type there is and what kind of a receptor the ligand binds to, meaning there are different types of cell membrane receptors. But the good news is that we can learn some generalizations without learning about all the different variations. We will learn that the extracellular signal is converted to an intracellular signal. That intracellular signal is called a second messenger, and it will either be an ion like calcium or sodium. It might be cyclic AMP, which is a form of broken down ATP, or it will be a kinase, which is an enzyme. The two most common are calcium and cyclic AMP. So those are the only ones we'll look at. What those second messengers do inside of the cell, what calcium and cyclic AMP do inside of the cell, will depend on the type of cell membrane receptor and what intracellular signal activated. To understand how a second messenger is even generated from a ligand molecule that's outside of a target cell, it's necessary to look more closely at some specific types of membrane receptors. And I chose just 3 types. They're the most common, and you will encounter them later in the course. In one type, called the chemically gated ion channel, the receptor is also a channel, and a specific ion can pass through that channel when it's open to get across the cell membrane. So when the ligand binds, the channel opens and the ion is allowed to diffuse in a direction consistent with its concentration gradient. So that's a chemically gated ion channel, which is a cell membrane receptor. The second type of cell membrane receptor would more specifically be called an enzymic receptor. In this case, the receptor also acts as an enzyme in that it catalyzes a chemical reaction in the target cell when it binds to a ligand. So in the picture, you see that double circle ligand, pink ligand, binding to its blue receptor, and that blue receptor is then active and catalyzes a chemical reaction. The last type of cell membrane receptor is called a G protein-linked receptor. And I know that's a mouthful, but it does explain exactly what its structure is. So this loop-de-loop protein through the membrane is the actual receptor that the ligand binds to. Next to it is a G protein. When the ligand binds to the receptor, the G protein is activated. Once this G protein is activated, it can move over slightly, and it will have an effect on any nearby cell membrane proteins. I think this will become a bit more tangible to you if we look at some very specific examples, and it'll help you to form your own generalizations about how G protein-linked receptor signaling works. So we're going to first, any kind of cell membrane receptor signaling. So first, let's go back to the chemically gated ion channel and look at a very specific example. The ligand in this case is acetylcholine, which is a neurotransmitter, and the nerve innervates a skeletal muscle. So the target cell is a skeletal muscle cell. It has an acetylcholine receptor that's a sodium channel. What will happen when acetylcholine, we'll assume this green molecule is acetylcholine, when acetylcholine binds to the blue receptor, this channel opens, allowing any sodium ions to actually move down a concentration gradient, which will be into the muscle cell. We'll learn a lot more about that when we discuss muscles and nerves later. But that sodium is necessary for a muscle cell to contract. Now let's look at an example of an enzymic cell membrane receptor. This occurs as your body is trying to lower blood glucose. And so I chose the ligand insulin, which many of us are familiar with. Insulin is a protein made by the pancreas that, of course, lowers our blood glucose after we eat. And it travels through the bloodstream to reach its target cells, and it has many target cells. We'll focus on skeletal muscle, but it also fat cells, adipose, and liver. The insulin receptor is what we're interested in. It's an enzyme, and it's shown as two different molecules because it occurs this way in your cells. It's considered what's known as a dimer, but you don't have to remember that. So the insulin receptor is an enzyme. This enzyme is also known as a kinase because what it does is it phosphorylates proteins. So what the action will be when the ligand binds to the insulin receptor. So this is insulin, the red balls. When insulin binds to the receptors, which are blue, this will become active, this kinase region will become active, and it will phosphorylate lots of proteins inside the cell. Those proteins will become activated, and they will have various functions. Specifically, we're going to see that one thing that will happen is that the cell will begin to store glucose as glycogen. That glucose will leave your blood, and it will go into the skeletal muscle and form glycogen. For now, let's just look at the mechanics of the insulin receptor one more time. In this diagram, insulin is green, the receptor is blue. When the insulin and receptor binding occurs, this receptor will phosphorylate, that's what the P's are, proteins inside the cytoplasm. So this red arrow shows a phosphorylated protein. Now this protein is active because it's been phosphorylated. What it will then do is activate other proteins, and one that it activates is called glycogen synthase, and that's what the green arrow is pointing to. Glycogen synthase takes glucose and turns it into a big chain of glucose is called glycogen, and that helps to lower blood glucose. What's important is that insulin is a hydrophilic signaling molecule that binds to a receptor that's an enzyme. Now, the last type of chemical signaling receptor that we're going to look at is called a G protein-linked receptor. The receptor is this ribbon that's winding through the membrane. The ligand are the two red balls. The G protein is this clump of globular proteins that becomes activated when the ligand binds. When this G protein becomes activated, it will facilitate the production of an intracellular signal by acting on neighboring proteins. This intracellular signal is called a second messenger, and the most common second messengers are cyclic AMP and calcium. They will cause a cellular response. The second messenger will. What's really fascinating, and maybe the most important feature of cell signaling that uses G protein-linked receptors, is that a very small amount of extracellular signal can generate a very large response. This feature is referred to as signal amplification. And if you look at this diagram, it tries to explain why this amplification occurs. We'll just look at a couple of steps. Notice that this ligand binds at step 1, and step 2 shows that the G protein is becoming activated. It's got a little pink blob on it, so I guess that's activated. Once it's activated, it moves off, and then another one can become activated, and then another G protein, and then another G protein. So that's one point of amplification right there. Furthermore, the G protein moves over to a nearby protein in the membrane. In this case, it's an enzyme. And as long as that G protein is bound, this protein can make many different products. And what we're interested in is the cyclic AMP. So lots of cyclic AMP will be generated. What cyclic AMP does is it will bind to other proteins and enzymes inside the cell to activate them, and you'll get a lot of change in metabolism intracellularly. So cyclic AMP is what we consider our second messenger. You could argue that as things cascade down, chemical reactions, all of these are second messengers. Cyclic AMP affects a protein kinase, which affects another enzyme. They're all second messengers, but we consider the cyclic AMP to be the first one. So since cyclic AMP and calcium are the most common second messengers, I'd like to explain how they're generated inside of a cell. So this is a G protein-linked receptor with cyclic AMP as a second messenger. The enzyme that the G protein will activate in this case is called adenylyl cyclase. You do not have to remember that. It's kind of pink. I tried to make a symbol here for you. The G protein will activate adenylyl cyclase, and adenylyl cyclase will convert ATP into cyclic AMP. Cyclic AMP is continually made as long as the G protein is bound to that enzyme, and that happens as long as the ligand, G proteins are activated as long as the ligand is bound. Cyclic AMP then binds to target proteins in the cytoplasm, which have various functions. Let's look now at how calcium is generated, how it becomes a second messenger. The first step's basically the same. This red triangular ligand binds to this purple receptor and that activates a nearby G protein, and then the G protein is going to affect a neighboring enzyme. But in this case, the enzyme is different, has a different name. It's called phospholipase C, not that you have to remember that, but it catalyzes a reaction that produces something known as IP3. Compound called PIP2 turns into IP3. You don't really have to know that. What's important is that that product opens the sarcoplasmic reticulum, which stores calcium. So calcium floods out, and calcium binds to target protein. Those target proteins, once bound to calcium, will be active, and they will perform many different chemical reactions. So I want to show you a specific example of a G protein linked receptor, just so you understand the relevance. And we'll do an activity in class too, so you're not too worried about this. But a G protein linked receptor is shown here in green. And in your body, real body experience here, your skeletal muscles use this in chemical signaling to respond to epinephrine. So the triangle in this case is epinephrine. That's the ligand. And when it binds to the receptor, the G protein is activated, activating adenylyl cyclase, which makes a lot of cyclic AMP, which activates a cascade of different proteins and enzymes. What's interesting about this is that cyclic AMP activates enzymes that cause your muscle cells to grow by making more protein fibers and by making those protein fibers thicker. So that's called hypertrophy. So epinephrine works through a G protein linked receptor to make your muscle cells bigger. So we covered a lot of information. I'd like to give you a brief summary of cell membrane receptor signaling. The important point is that an extracellular ligand is converted to an intracellular signal known as a second messenger. That extracellular signal is amplified to become stronger because the G protein is activated as long as the ligand binds, and there are multiple downstream components that amplify at multiple steps. The cellular responses are very diverse. Some examples that we've looked at is metabolism changes, cellular growth. We haven't looked at it, but a cell could also be caused to secrete a substance in response to cell membrane receptor signaling, specifically G proteins. Thank you.